I think it will be turned on. Shall I go up or? Yeah? Okay. Yeah, I have a laser pointer, yeah. But I also have a lot of animation, so I think for the YouTube people that will be fine, yeah. Is it okay if I use the laser sometime? Okay. Yeah, I have it all set up.
Bueno, buenas tardes. Muchas gracias por estar aquí. Gracias también a las personas que ya se conectaron en el canal de YouTube. De verdad que es un gusto poder ver el auditorio así. Y es un placer para mí presentarles a la doctora Marieke Kuijjer de la Universidad de Oslo. La doctora Marieke obtuvo su doctorado en la Universidad de Leiden, en Holanda, y su entrenamiento postdoctoral en el Dana-Farber Cancer Institute en Estados Unidos. Y desde el 2018 tiene su laboratorio, obtuvo el nombramiento de jefe de grupo en el Centro de Medicina Molecular de Noruega en la Universidad de Oslo. También mantiene un appointment. I think you have an appointment in Netherlands, right?
Yes.
Okay. La doctora Kuijjer ha publicado 57 artículos, los cuales han recibido alrededor de 3.300 citas. También ha publicado 12 softwares para el análisis de datos masivos. Ha formado estudiantes de maestría y de doctorado, y en su laboratorio se estudian los mecanismos moleculares que participan en el desarrollo, progresión y heterogeneidad del cáncer. Para ello, su grupo desarrolla herramientas computacionales para analizar redes de interacción genómica y mediante inteligencia artificial mapea paisajes regulatorios génicos de pacientes con cáncer. Y bueno, hoy nos va a presentar algo de su trabajo. Tengo que aclarar también que la doctora Marieke está muy abierta a colaboraciones. Si alguno de ustedes, después de escucharla, quisiera hablar con ella, pues nada más se pone en contacto conmigo y organizamos su agenda. Ella va a estar hoy y mañana, día completos, y el miércoles en la mañana. Marieke, thank you for being here. Thank you.
Thank you very much, Leonor, for the really nice introduction. I got a little bit of it, so I think I don't have to introduce anymore what my lab is working on. But yeah, and thanks also very much for the invitation to be here. It's really beautiful to be here in Cuernavaca, and I'm really happy that I could visit in person. So this talk is going to be a bit similar to, so I gave a talk about a year ago at the Frontiers in Genomics program. So for those who attended it, there will be a bit of overlap, quite a bit. But I hope, it will help as a refresher of what I presented then. So basically, what I would like to show you today is the work that my group is focusing on. It's mostly both the development of computational tools to model genome-wide gene regulatory networks, and then also the way to use these networks to analyze how networks can influence disease. We mostly focus on cancer. So the examples I'm going to show you today will be in cancer, but just so you know, the tools that we develop can also be applied to other systems. And we have some collaborations, for example, in mice as well as in plants. So to give you a bit of an idea of why I believe it's important to model networks in cancer, I just wanted to give you this very small example of two women who come to the doctor with some complaints. They have different genotypes. They might have different working environments, and they might also have different diets. And unfortunately, both of these women develop glioblastoma. This is a very aggressive form of brain cancer that I'll be talking about today. They're both treated with surgery, radiotherapy, and also chemotherapy, so very heavy treatments. But while Sarah's tumor responds well to these treatments, Jane's tumor doesn't respond very well. So with precision medicine, what we hope to do is take into account all these different data types to better understand or to get an idea of why, for example, Sarah tumor responds, but Jane's doesn't. And I hope that today I can argue that we also need to model gene regulation, in particular genome-wide networks, to get a better idea of what is happening in the tumor. So this is an overview of my talk. I'll start with a general introduction on gene regulatory networks, what they are and how we can model them. And then I'll show you a couple of tools that we've collaborated on as well as developed in the group that all have animal names. So I'm going to talk about Panda and Puma. These are two tools that can be used to model transcriptional networks and post-transcriptional networks. And I will be showing you some applications in healthy tissues to better understand tissue-specific gene regulation. Then I will be introducing a tool that we call Lioness. This is an approach that can be used to model networks for individual samples or individual patients. And then after having introduced how these methods work, I will show you an application in a disease, in specific in glioblastoma. And I'll end with some current and future directions of the lab. We are contributing to something that we call the Network Zoo. This is a collaboration with several groups on the work in the world, driven by the John Quackenbush group, where I did my postdoctoral work. And we basically develop different tools to model different types of networks and also to analyze them. And so Panda, Puma, and Lioness are part of this Network Zoo. And I'll also introduce some other newer tools that my group has been developing as part of the Network Zoo. So let's start with gene regulatory networks. I believe it's very important to model gene regulation in biology. Of course, it's known that human cells or other cells carry out many common processes, such as those that define their survival or cellular structure. But cells also exhibit these unique functions that can help define their phenotypes. So our cells have the same DNA, but different tissues can arise from this exact same DNA. This is really driven by gene regulatory processes. They define which genes are expressed, and they also control the timing and the extent of the expression. And so here you can think of transcriptional regulation, where you might have a transcription factor that binds to so-called transcription factor binding sites in regulatory regions, such as the promoter of a gene, and thereby influences its expression. But we can also model or look into post-transcriptional regulation driven by, for example, microRNAs that can bind to mRNA transcripts and thereby downregulate them, or even RNA binding proteins that can influence, for example, isoform expression levels. Now, together, these regulatory elements form a complex network of interactions between the regulators, so the transcription factors or the microRNAs, and their target genes. And these networks are thought to be different, for example, for different tissues. But what I'm mostly interested in is actually understanding how disruption or rewiring of these networks can drive cancer or these other types of diseases. In cancer, you can, for example, think of one node, a transcription factor here, that might be overexpressed. If it's overexpressed, it might have maybe a higher chance of binding to binding sites and thereby, for example, influence the expression of an oncogene. But we can also have a modification of an edge or interaction in these networks, shown over here. This can be, for example, a mutation in a transcription factor binding site so that a transcription factor cannot bind anymore. And this may, for example, lead to the downregulation of a tumor suppressor gene. So how do we actually model these networks? The ideal way of modeling a genome-wide network is to take into account all transcription factors or all target genes in the cell. And then if we know that regulation is happening, we would draw an edge in this network. And we would do this for each possible regulator target gene pair. Now, there's several ways of actually getting to these networks. One is through experimental approaches. So, for example, for transcription factors, you can think of ChIP-seq experiments, where you look where does the transcription factor actually bind to the DNA. These are really nice experiments to do, but the difficulty is it's really hard to do them genome-wide. So the human genome has over 1,000 transcription factors. And if you would have to do one experiment for each of these transcription factors, it's going to be very expensive. And it also needs lots of material. So often you need hundreds of thousands of cells to do this. And if you, for example, want to look into small tumor biopsies, which is what my group is interested in, it will be really difficult to model these networks with experiments. So we use computational network inference to do this. And to give you a bit of an idea of how computational network inference is often done, I just wanted to show you two examples before actually showing what we use in my group. So often people look at correlations between the regulator, for example, the transcription factor, and the target gene. And then the idea is if these two, if the transcription factor and the target gene are co-expressed, so which you can see over here, there's likely regulation happening. Other approaches start with some prior knowledge, for example, on transcription factor binding. So they start, for example, with a prior network where you have all transcription factors and their transcription factor binding sites, and then color these networks with something that we call network painting, based on the expression levels of the transcription factors in the target genes. The problem with these approaches is that we and others have found that correlation doesn't necessarily mean regulation. And an approach such as this network painting is based on some fixed initial network that cannot change. You can only change this network by coloring the nodes, but you cannot identify any possible new edges in the network. So we are working with an approach that uses message passing. It's called PANDA, and I will show you not the next slide, but after that, how it works. But before that, I just wanted to give you kind of an idea of what I mean by message passing. So to explain this, we have an example of a very small classroom. We have one teacher, and then we have two students. And this teacher is trying to explain an equation to these students. Now, if the teacher doesn't do a very good job here, the students will probably not understand this equation, and they will likely fail their exams. Well, if the teacher knows how to explain this well, the students will probably get better grades. So this teacher is responsible for teaching. Students, however, also need to be available to learn. So if they're not paying attention, they will probably get worse grades. So responsibility and availability are two terms from message passing. And we can apply this to our networks as well. In our case, the transcription factor is the teacher. The transcription factor is trying to send a message to its target gene. And then the genes, the target genes, are the students. They're trying to receive a message from the teacher. Now, the great thing about the genome-wide networks that we are looking into is that we do not just have one teacher and two students, but we actually have a whole school with different teachers and lots of different students. So about 1,000 transcription factors and about 20,000 target genes in case of the human genome, where we can look into, for example, how does the performance of different teachers compare to one another, or how does the performance of different students compare to one another? And we can use this approach to check where the message passing works and where the message passing fails. So this brings me to these methods, PANDA and PUMA, that we can use to model gene regulatory networks. This is going to be a very dense slide, so I will spend a bit of time on this one, but I think it's important to get a bit of an understanding of how this message passing can be applied before we actually start applying it. I will first explain PANDA. This was an approach that was developed by my colleague, Kimberly Glass from Harvard Medical School. It starts with some regulatory prior. So it starts with some initial information on potential transcription factor binding. For example, obtained through looking into these motifs of transcription factors and where do they bind to promoter regions of their target genes. It then integrates this with protein-protein interactions between transcription factors, as well as gene expression data, for example, obtained from microRNA or RNA-Seq data. Now, the first step is this expression data is going to be converted into co-expression. And this is important because we want to check the performance, we want to compare the performance of the students to one another, so the performance of the target genes. So we start off with 3 initial networks. And we're then calculating the responsibility and the availability. Now, to explain this, to get a bit of an intuition on this, it works as follows. So, for example, we have these 2 transcription factors that from the protein-protein interaction data are known to be able to bind together. And if we know from the regulatory prior that this first transcription factor can bind to the promoter of this specific target gene, we might increase the evidence a bit that the other transcription factor does the same. Why? Transcription factors often form complexes and work together to influence the target gene expression. The same holds for the availability. If we know that 2 genes from the co-expression data are being co-expressed, so they're always over or under-expressed together, and we know from this regulatory prior that the transcription factor regulates the first target gene, we can increase the evidence a bit that it also regulates the other target gene. We can use this information to update this initial network. So we just change these network interactions a bit based on the information we obtain from the responsibility and the availability. We can do the same then with the other two types of networks and loop through this many, many times. So this is an iterative process. At some point, this converges, and we then obtain one network, the network that has found agreement between these three different data types. Now, importantly, message passing looks at overlap in targeting patterns between these three different input networks. But transcription factor expression levels are not used to model interactions. And also this approach can learn potential new interactions. For example, if you have a mutation in a transcription factor binding site. Puma is based on Panda. It's an adaptation of Panda. It's actually very, very similar. We use microRNA target predictions in this regulatory prior, and it then has an adapted message passing approach. We adapted the responsibility. I'm not going into detail here, but I'd be very happy to answer any questions if you have those. And the output of these two different methods are very, very big networks. So we get interactions between each regulator and target gene pair. So these networks are very, very big. We often work with, the examples that I'm going to show you today, work with about 600 transcription factors and about 20,000 target genes. So we often have networks that are 10 to 20 million edge weights. And these edges are weighted, and these weights represent likelihoods of regulation. So the higher the edge weight, the more likely it is that there's regulation between the transcription factor target gene pair. And finally, this approach, or both Panda and Puma, model what we call an aggregate network. We have some input population, for example, a cancer data set. We apply this whole approach, and we just get one network. And I'll come back to that later. So now let's apply these approaches to healthy tissues. Here we wanted to analyze data from the Genotype Tissue Expression Project. And this is a very large data set that contains over 10,000 individuals, and then gene expression data derived from different tissues. We decided to model networks for 38 different tissues based on the gene expression data, integrating it with Panda, with protein-protein interactions and this motif prior. So we used Panda to model 38 different networks. And what we then wanted to do is we wanted to see, can we actually look into these networks to see, are there any differences between each of these different tissues? And we wanted to look both at the edges in the network, so the interactions, and at the tissue-specific nodes, so the transcription factors and their genes. Now, to compare these 38 networks with one another, we decided to look in a very simple metric, which we called a tissue specificity score. So basically here we have a network, say a network for brain. If an edge weight is very high in this network, we compare it to the edge weights across all 38 different networks. And if this edge weight is very high and much higher than the edge weight over all 38 different networks, we call this edge tissue specific. We base this on something that's called an outlier statistic, so very simple. Now, combining this with the gene expression levels of the transcription factors and target genes, we found something quite interesting. What we did is we looked into edge multiplicity. I won't go into too much detail here, but this is basically an edge multiplicity of 1 means that the edge is only specific in the tissue we're interested in. An edge multiplicity of 2 means it's specific in 2 tissues, etc. And this can happen because some tissues are very similar. For example, we had here in this specific data set, we had 3 different tissues derived from the brain. So there will be some tissue-specific interactions that are specific to brain. So they will then be specific in 3 different brain networks. What we found by combining this with the expression data is that in general, if a gene is specifically expressed in the tissue, it also has tissue-specific interactions, and that can be shown over here. So this is basically the enrichment in tissue-specific genes of these specific edges. But we found that this was the case too for transcription factors, but this effect size was much, much lower. And this indicates that transcription factors do not necessarily have to be differentially expressed to regulate their target genes. So it can happen that a transcription factor is stably expressed across all 38 different tissues, but it can still regulate their target genes in a tissue-specific manner. We also wanted to look into this into a bit further detail, and we decided to look into all the different interactions that were specific in a tissue and that were attached to a specific transcription factor. So here I'm just drawing this network, this brain network, in what we call an adjacency matrix, where we have the transcription factors on the x-axis and then the target gene on the y-axis. And so say we are interested in one transcription factor, we can take all of these interactions or edge weights that are connected to the transcription factor and then compare it to the same interactions in all the other 38 different tissues. We can then do gene set enrichment analysis to see, are there any specific biological pathways that are enriched in this specific tissue targeted by this specific transcription factor. We call this tissue-specific transcription factor targeting profiles. And we did this for each of the 38 tissues and for all of the 650 transcription factors. So this was a very big analysis. I'm just going to show a very small example where we looked into brain. And what we visualized here is basically those pathways that were enriched in brain that were connected to the 10 transcription factors that were least specifically expressed to brain. So they were kind of stably expressed across all tissues. What we saw, so here in red, you see enrichment of these specific pathways. And these pathways play a known role in brain, such as neurotransmitter secretion or the regulation of neurotransmitter levels. So based on this network analysis, we could actually identify interesting transcription factors that we couldn't find based on their gene expression levels alone, but that do regulate these interesting pathways in brain. Which we came to the conclusion that it's possible with these networks to now highlight specific transcription factors as well as gene, just by placing them into the framework of a network. We also applied this PUMA approach to the same data set. So we again had these 38 different tissues, and then we wanted to model networks based on microRNA gene regulation. And to do this, we decided to start with two different prior networks. One was based on microRNA target predictions from TargetScan, a very well-known resource, and the other for Miranda. So these are two of the widely known resources for target predictions. Now, the problem with microRNA target predictions is that these are often not very robust. So there's quite some differences between these two different data sets. And we wanted to be sure, can we, with our PUMA methods, model networks that are robust, even though these two different input data sets are very different from one another? So we modeled two collections of networks based on microRNAs for the 38 different tissues. And then decided to compare these tissue specificity scores that I explained to you in the previous slide. And this is a visualization of the number of tissue-specific interactions we find between microRNAs and their target genes for the 38 different tissues. I won't go too much into detail here. We see some different numbers of microRNA target gene interactions between the different tissues. What's striking here was that these numbers are very similar, even if you model these networks on these different input data sets. We also calculated these targeting profiles now for microRNAs and looked into the similarity of the targeting profiles that were modeled on this TargetScan and this Miranda input data sets. And this figure shows a Pearson correlation, so similarity between these two different data sets, which we found to be generally quite high. So it seems that the PUMA approach can consistently predict tissue-specific functions of microRNAs. We actually put these functions in a database that can be accessed online. I'm sorry I didn't put the link here, but this is a Shiny app. And if you're interested, please take a look at the paper. The reference is over here. Finally, we wanted to see, can these networks potentially highlight disease associations? And to do this, we decided to integrate the data with a database that's called NERD-SNP, that basically has single nucleotide polymorphisms in microRNA binding sites. Now, what we did was we we checked, first of all, in which genes these SNPs were available in the specific tissue that was associated with the disease. And then we compared the function of these genes with the function of the microRNA predicted tissue-specific functions from PUMA. And we saw that we could capture for about 86% of the genes, we could capture similar functions. So this means that with an approach such as PUMA, we can potentially identify disease associations. So this is nice. I showed you some examples of how we can use these aggregate network models to study tissue-specific gene regulation. But what happens in cancer? In cancer, we really would like to model networks for individual patients. So this brings me to the Linus approach, which we developed to do so. So a little bit more details on why I believe it's important to model networks for individual patients. So the problem with, for example, cancer is we can have maybe two data sets, maybe a cancer data set and a control data set. Then use methods such as PUMA or PANDA to model networks. Then we get a cancer network and a control network. But we cannot do any statistical comparisons between these networks. We can just look at the effect sizes of these edge weights between the two. Also, with cancer, it's very important to model what's happening in the individual patient because cancer is a very heterogeneous disease. Now, we developed this Linus approach, but when we did so, there was actually already what we call a workaround to model tissue-specific networks, which was basically take some expression data, model an aggregate network, maybe with PANDA, and then color the edges or the nodes, so the transcription factors and their target genes, based on the expression levels in each individual samples. But we didn't really like this so much. First of all, because from the GTEC studies, we found that a regulator's expression level is not necessarily correlated with its target genes. And also this approach, again, isn't able to model any new edges that might be specific to one specific patient. So together with Kimberly Glass, I worked on an approach that we call Linus that is actually very simple. So here we have an input data set of, for example, cancer patients. We have some expression data integrated with some other data types with PANDA, and then we get one network that is estimated using all samples in the population. We asked ourselves, what happens if you remove one specific patient from the input data set, model the network again, and then we have a network estimated using all samples except this specific patient. It turns out that removing a specific patient from this input population data set perturbs the network. We can check these perturbations, and these are the contribution of this individual sample to the aggregate network model. We have to normalize this, and we actually normalize it by multiplying this by the number of input samples in the data set. You can imagine if you model a network on 10,000 samples and remove one sample, model the network again on 9,999, these differences are going to be really, really, really small. Well, if you model the network on 10 samples, remove one sample, model the network again on 9 samples, the differences are going to be much bigger. So we multiply these differences with a scaling factor, add it back up to the network modeled without a sample of interest to get the individual's network. We can actually show that this works with simple network reconstruction algorithms such as Pearson correlation. And in the Linus paper, we also showed that it works with more complex methods such as PANDA. I won't go into detail here, but we tested the Linus approach in different data sets, in silico data from yeasts that were cycling through the cell cycle, and then also human cell line data. And we found that the networks were reproducible, they were also accurate, and they were also biologically meaningful. Now, since developing this, so this was published in 2019, we have applied it already to a couple of different data sets, including the GTEx data set, where we looked into sex-specific differences of individuals. But I just wanted to quickly mention that this approach is not limited to regulatory networks, so you don't have to necessarily apply it to PANDA or PUMA. It has been, for example, used also to look into microbiota co-occurrences by other researchers. Now let's apply Linus in disease. So a little bit of more background in cancer. I already showed this in the beginning, that cancer is a very complex disease where multiple factors play a role. It's also very heterogeneous. So we often find different patient subtypes that may be defined by specific signaling pathways that are active or that may be associated with survival. Now, many different studies have been already looking into different omics landscapes, such as gene expression data, copy number changes, or methylation in cancer. And while this has helped our understanding of cancer, it also found that most cancers are really very complex. And for a large fraction of cancer patients, we cannot always point to specific driver genes. So genes that drive the cause of cancer or, for example, poor survival. So our thoughts here was that aberrations in these regulatory interactions can also drive cancer. And we should model these interactions genome-wide, and this can be done with approaches such as PANDA and Lioness. Glioblastoma is a very aggressive form of brain cancer that has very, very poor overall survival profiles. So this was one of the reasons why we wanted to specifically look into this disease. The overall survival of patients is very poor. So in about 5 years, most patients have unfortunately died. So we were wondering what specific molecular features drive this poor survival. And at the time when we started this study, there were already quite some signatures published on gene expression data. So basically, you look into some gene expression, try to associate it with survival, and then check which are the genes that contribute to this. But what was very striking here was that all these different signatures that had been published included different genes, so there was not much consistency there. So we were wondering, could gene regulation maybe play a role? So we started modeling networks. We had protein-protein interaction data for the transcription factors that we obtained from publicly available data. We started with what this was a regulatory prior of potential transcription factor target gene interactions by looking at where do specific transcription factor motifs match to promoters of the target genes. And then we had 2 different expression data sets available from the Cancer Genome Atlas, a very big resource of cancer data. So these were 2 different data sets. They were modeled on 2 different microarray platforms. And we call this Discovery Dataset 1 and Discovery Dataset 2. So we used PANDA and Lioness to model networks for individual patients. So then we got 2 collections of networks, one for the first discovery dataset and the other for the second discovery dataset. And for simplicity, let's just focus on this one for now. So what we then did is we divided this patient population into 2 groups. One group of patients that had relatively long-term survival. So they survived at least 20 months after initial diagnosis of the primary tumor. And then the other group was the short-term survivor group where patients died within 20 months of the diagnosis of the primary tumor. We then wanted to compare these 2 different sets of networks with statistical approaches. But I already showed you that these networks are very, very big and that they're a bit tricky to analyze. So we started very, very simple and wanted to analyze the networks on the gene level. We calculated what we call gene targeting scores. This basically looks at all the interactions that point to a specific gene and then sums them up. And what we could then do with the gene targeting scores is compare them between the 2 different survivor groups with a statistical test. For this, we used LIMA. It's an approach that was developed to analyze expression data, but it turns out to work out quite well on our networks as well. And the nice thing about the single sample networks is that we could now also correct this analysis for covariates, such as the patient's sex or the patient's age. And now that we had these scores for each individual gene, we could look into gene set enrichment. So we could check, are there any specific biological pathways that are enriched for differential targeting? We did this for the 2 discovery data sets. So here in this figure, you see the enrichment scores from the gene set enrichment analysis. And then the significance is the color of these bubbles. And then the 2 bubbles show the 2 different data sets. Actually not very important because this is all significant. What we found is we found 7 pathways that were significant in both data sets. And these pathways all played a role in the immune system, so that got us quite interested. But we weren't very sure if this analysis was robust. So we decided to then repeat it in some independent data sets. We had a data set from the German glioma network and also a data set from Japan, where we basically repeated this entire analysis. And we could then capture one of these pathways, PD-1 signaling. Looking at the network of interactions around genes in the PD-1 pathway, we saw that this what's likely happening here is repression of the genes in this pathway in the long-term survival group. So here we show the genes that are overexpressed in the short-term survival group are shown in red. And then the edges, so the edges that are high in the long-term survival group are shown in blue. So this likely indicates repression. And this got us very interested because the PD-1 signaling pathway is important in cancer. Cancer cells can express certain proteins that can then bind to T cells and can inactivate them. And then the tumor cell can escape the immune system. But now there are specific immunotherapy options available to revert this. We obviously wanted to make sure that our findings were not associated with differences in different cell types that are present in these tumors. So we did what's called deconvolution analysis to look into specific immune components in these tumors as well. And we found that there was no true significant difference in terms of how many T cells or other cells are infiltrated in these tumors between the long-term and short-term survival group. So it seems that the PD-1 pathway repression is an independent prognostic biomarker in glioblastoma. And what we hope now is that potentially, and this is obviously still very far from the clinic, but potentially in the future, what we could do is try to inhibit these transcription factors, to target them, to revert the PD-1 signature to its repressive state. So with this, I hope to have kind of convinced you that we need networks to better understand what's going wrong in these different tumors. And potentially by now altering these networks in the cancer patients, we might be able to help them and to help them improve their survival rates. So let's focus a bit on the conclusions and future directions of the group. So I believe that to better understand cancer and actually other complex diseases as well, it's very important to shift from analyzing these single data types towards modeling these complex interactions. I showed you two approaches, PANDA and PUMA. PANDA models transcriptional networks and PUMA models post-transcriptional regulation by microRNAs. Then I showed you the approach we call Lioness that models networks for individual patients and that now allows us to statistically analyze these networks. And by combining these approaches and applying them to data from glioblastoma, we found that loss of repression of the PD-1 signaling pathway is associated with poor survival. So what we try to do in the group is really look into these complex regulatory interactions. We focus mostly on transcriptional regulation, but we also look a bit into post-transcriptional regulation by modeling these condition-specific or aggregate networks and check if there's any changes in these networks, can it drive disease? We also use these single sample network approaches to connect this to precision network medicine, for example, by integrating our networks with clinical data or other data types, to identify new cancer subtypes, or for example, to check new ideas or to get new ideas on potential response to target treatment options. We focus on developing tools to improve gene regulatory network modeling, new network analysis approaches and applications of those mostly to cancer. And obviously with the recent burst in single cell and spatial transcriptomic methods, we're also interested in further developing these tools towards these new different data types. So I just wanted to highlight very shortly some of the recent work we've done in each of these three different directions. The first one really focuses on improving the input data for network modeling. So we actually found by looking into this data, this was mostly by looking into data from GTEx, which was very heterogeneous, that different normalization methods actually can influence the networks you get from them. We specifically found that quantile normalization-based approaches can introduce errors in the networks, and that's obviously something we don't want to have. And this is mostly or actually only in RNA-Seq data, where you have a lot of zero counts. And the problem with these zero counts is that they get tight values, and then they basically, the normalization just gives a specific number, adds a specific number up to these zero counts. And then what we then get, if we look into co-expression data, we see that there's a lot of false positive associations. So here, I'm sorry it's not very visible, but here we looked into specific tissues, again from the GTEx data set, and we looked into genes that were exclusively expressed in those tissues, so that didn't have any signal in the other tissues. And we observed lots of co-expression that shouldn't be there. So Ping Han, a PhD student in the group, developed an approach that we call SNAIL. It's an adaptation of the smooth quantum normalization methods that was developed a few years ago that allows us to normalize very heterogeneous data sets, such as the data from GTEx. But it uses non-parametric statistics to remove these false positives to form. And we tested it in different data sets, both from GTEx and also from ENCODE, and found that it is quite effective in removing these false positives. Now, for the second part, we are also working towards getting a more fine-tuned network analysis. So I've shown you a very, very simple approach where we looked into these gene targeting scores. So we basically summarize the networks and just look at what's happening at the level of the gene. But we were wondering, can we actually move from these summary statistics or from these gene targeting scores towards analyzing the complete network of 10 to 20 million edges? We also wanted in cancer to move from discrete subtypes towards mapping more the subtle heterogeneity across patients. So often in cancer, people like to identify 2 or 3 subtypes that might associate with some clinical features. But we thought actually looking into the heterogeneity as a spectrum is a much more realistic view of what's happening in cancer. So Tatjana, a postdoc in the group, developed a tool that basically makes use of these different ideas. And what it does is it incorporates our knowledge on biology to find heterogeneously dysregulated pathways. We call this tool Porcupine. And Porcupine makes use of dimensionality reduction, so principal component analysis to do so. So it is a very quick explanation on PCA. You've probably all heard of this, but it basically is an approach to try to look into the data and explain it with fewer features. So you look at what are the most important principal components or features that drive heterogeneity, in our case, in our specific networks. And one thing that's really useful of this analysis is that it gives you some idea of how much the data can actually be explained by these different features. So Tatjana decided to use this idea of principal component analysis and also known biological pathways to look into our networks. And it works as follows. So here I'm just showing you 2 different pathways, pathway 1 and pathway N, that contain different genes. Now what we can do is we can look at the edges that are connected to these genes. So here we have the transcription factors and then the genes. And it turns out we can do this for individual patients, and you can see here, there's quite some heterogeneity. So there's some differential regulation in this specific pathway in this data set. While here, for pathway N, we find only a little bit of variation. We map this variation with principal component analysis. We look into the first component that explains most variance, and then compare this to a random background to check, is this signal actually significant? And then what we can do is we can say, well, this here, for this specific data set, there's lots of variability in this pathway. So this pathway shows significant differential regulation in the data. Well, for pathway N, this is not the case. Tatjana applied this to soft tissue sarcomas. I won't go much into detail here either, but this is basically a cancer type of mesenchymal origin that is super heterogeneous. So it can derive in different tissues. And she specifically looked into soft tissue sarcoma, mostly due to data availability in the disease. She modeled networks for all of these different soft tissue sarcomas, and then placed these on a UMAP. So basically looked at the variability of these networks across all the different patients. And here different colors mean different tumor types. I'll just want to highlight this one. So these blue and green samples are leiomyosarcoma. It's a sarcoma type that can arise from smooth muscle tissue. And what we found here is that these leiomyosarcomas were very heterogeneous. So we see potentially 2 subtypes, although it's a bit hard to say if these are actually 2 subtypes. So we decided to apply Porcupine to this data set. And then we obtained some pathways that were significantly dysregulated across the patient population. And here on the x-axis, we show the enrichment scores from the gene set enrichment analysis from the Cancer Genome Atlas, so one of the data types that we had. And then on the y-axis, we showed the exact same analysis, but then applied to another data set from our collaborators in Germany. We identified 37 pathways that were differentially regulated. These pathways correlated quite well, so it seems that this analysis was robust, at least between these two different data sets. And what we can now do with this, and this is what I think is kind of our next step, is look into an individual pathway and then check, is this pathway enriched for specific transcription factors? We did this for the 37 pathways. We found one transcription factor that seems to be highly active in differentially regulating its target genes, E2F8. And then we also looked into, try to validate these findings in data from leiomyosarcoma cell lines. So we had data available for actually only 4 leiomyosarcoma cell lines. So this is a very rare disease, and it's really difficult to get to get data here. But what we decided to do with these cell lines is look into the RNA-Seq, model the networks, map them on this map of patient data. And what we saw is that these cell lines are actually also quite heterogeneous in terms of what is being regulated. And then we decided to look into chromatin states with a tag-seq. And we found that all the genes from these pathways in general were more in open chromatin states. So we thought they're more likely to be dysregulated or heterogeneously regulated than other genes. And we could also find for the top pathway, this one, that there is also a differential regulation between the 4 different cell lines, indicating that the chromatin states are important in driving this heterogeneity that we obtain from our networks. The last example I wanted to show you is the example in single cells. This is work done by Daniel Osorio. He was a former postdoc in the group and is now at Boston's Children's Hospital. Daniel basically wanted to expand PANDA approach towards single cell data. So this figure looks a bit different, but it actually just looks very similar to the PANDA framework, where we have 3 different input data sets: protein-protein interactions, the transcription factor motif prior, and then single-cell gene expression data. And then we use this responsibility and availability from the message passing approach in PANDA to model a gene regulatory network. But this is a different step that Daniel applied. It's called supercells, and it's an approach that was published not too long ago in BMC Bioinformatics by another group, where they basically try to take expression data from single cells and then match cells that have very similar expression profiles together. And this kind of disparsifies the data a bit, so it makes it a bit easier to model networks on these data types. Now, we tested this approach actually in very different settings. So Daniel did a very large benchmarking study where he compared it with other single-cell network methods. I won't go into detail here, but it seems to work quite well. It's fast, and it seems to be able to capture actual regulation. And one of the examples that Daniel worked on that I really like is single-cell data where we had, this was from publicly available data sets, where we had either overexpression or knockout of specific transcription factors. And we could compare the expression levels in each individual cell with its targeting in each individual network that we modeled. And we found that, so for transcription factor, this transcription factor was overexpressed. We found that the targeting of this transcription factor was generally also higher. So we generally observed very high targeting scores in our networks. And then from the transcription factor that was knocked out, these targeting scores were generally quite low. So this is kind of the first example that we have. We're currently still working on the paper, but we also have some applications of this approach to single-cell data from colon cancer. Now, before ending, I just wanted to highlight a bit of the work from the Quackenbush group. Marouane Ben-Gabila, a postdoc in the Quackenbush group at Harvard, is very, very active in both contributing a framework for hosting all these different network tools, which we call NetZoo. So if you're interested in using any of these approaches, please take a look at the NetZoo GitHub. We also have netbooks or notebooks, Jupyter notebooks that are available that can be used to kind of reproduce some analysis. So if you're interested in reproducing, for example, the analysis we've shown in glioblastoma, that's possible. You can just go to this link. I believe you need a GitHub account for it, but then you can basically just reproduce the analysis and also reuse the code for your own projects. And then we have this a large collection of networks that we call Grant that is very useful for us to host our networks, so to make our networks available to the community. And it has various networks in there. So both networks on cancer and also some of the tissue networks that I've shown you in this talk. I showed you many different animals. These are all the tools that were developed in my group. Many of them are available in different programming languages in this net zoo. And then I show you a couple of new tools that were currently either on the bioarchive, such as snail and porcupine, or that are still in preparation, such as scorpion. We actually also have a tool that we call Retriever that can be used to recommend drugs based on single cell data, so could be also of interest. And these are available still on the group's GitHub, and they will be ported to NetZoo very soon. So with this, I'd like to end. I'm very grateful to my group over here. I showed you some work today of Tatjana and Daniel, a postdoc and former postdoc in the group. Pinghan, a PhD student in the group. Tess was actually a student that worked with me on the glioblastoma project, together with several people from the Quackenbush group, including Camila Lopez Ramos. And then Kimberly Glass and Abhijit Sanawane worked with me on the GTEx analyses. And I'm also very grateful to John Quackenbush and also to our collaborators in Germany who helped us with the validation experiments for the porcupine project. So thank you, and I'm happy to answer any questions.
¿Preguntas?
Hello. I have a question about when you look at the glioblastoma data, for instance, do you see different networks depending on the age of the tumors? And do you see something like a progression? Because it's very striking to see such an effect on longevity. Is it because it's an older tumor or something like that?
So what we actually did here is we specifically removed the age effect. So we controlled for that in our analysis. So with this specific analysis that I've shown you, it's not possible to see that. But actually some of the postdocs in John Quackenbush's groups are very interested in that. And they're actually looking into this now to see if we can use the networks and kind of use them as a proxy for age. But I don't have an answer, unfortunately.
Thank you.
Thank you. ¿Alguna otra pregunta?
Hello, thank you for the nice talk. I have a question, just more a curiosity question regarding the analysis of transcription factors versus microRNAs. Would you say, Mike, what analysis gives you a better kind of separation between different tissue types? Are microRNAs better for that, or transcription factors? Obviously, both are probably the best.
Yeah, so in terms of separation for the tissue types, I'm not 100% sure. But in general, I would say transcription factors. I cannot tell you if this is because of biology or because of the way we model these networks. Because with the transcription factor networks, we also use the protein-protein interactions between the transcription factors. So we have a bit more things to look into. So it might be due to how the algorithms are designed. That said, I think in general, transcription factors have more influence on the target gene expression levels. MicroRNAs are supposed to have a bit more subtle influence of how they change the mRNA transcripts. Yeah. Does that answer your question?
Yes, yes, thank you.
Hello, thank you for your talk. I have a doubt. If you make individual networks for every individual with cancer, you can tell if there are different transcription factors that drives to the same type of cancer.
Yeah, that's possible. Yeah, although it depends a bit on how you want to use the network to analyze. But what you can do is you can look into these targeting scores of the transcription factors and then look into enrichment. For example, with the porcupine approach, you can look into whether there's specific transcription factors that are highly enriched in driving heterogeneity. So that is possible.
Thank you.
Does it make sense? Yeah.
Hi, thanks for your presentation. It was very nice. My question is about, have you tried to do this kind of analysis contemplating other kind of non-coding RNAs, like long non-coding RNAs, or are you in plans to do a new algorithm for them?
So we haven't looked into that in specific, and I don't have any specific plans. I do think it's very interesting, but I don't have much expertise on the long non-coding RNA. So what you need for this is good prior information that have some idea of whether a long non-coding RNA can influence its target. Mm-hmm. And I'm not really sure where to get that, but it should be possible to apply it. But no, yeah, we have so many things we're already working on.
Thank you.
Thank you. I have a question about the lioness analysis. Are there any topological features that can be associated to outcomes, to each patient, individual outcomes on the patients?
So you mean topological network features?
Yeah,
very interesting. We haven't applied this with lioness. It's a bit, so basically, some of the tools in the NetZoo can look into network topology and can also look into differential network topology. And so this is work by Megha Padi. She's working at the University of Arizona in the US. So she developed an approach that we call Alpaca that can look into this differential topology. I think she is working on applying this now for single sample lioness networks, but I'm not sure where that's at. I think that's not yet available. It's a bit tricky because network topology, to kind of get good network communities, that's very computationally intense. And it's kind of easy to do that for cancer and control. And so Megha has published a bit of work on that, or for different cancer subgroups. But for single samples, it's a bit more tricky. But it would be super interesting to look into, yeah.
Hello. We have a couple of questions that were asked through our live chat on YouTube. The first one is about what would be the approach to studying external gene interaction, external gene regulators in a disease? For example, the interaction of DNA and transcription factors from a hepatitis V virus in a hepatocyte.
Oof, that's a tough one. I'm not sure. It's a bit outside of what I'm working with. So can you say again the example?
The example was about the interaction between viral DNA and virally originated transcription factors. So as opposed to your cancer analysis, where it's a disruption of a pre-existing network, what would happen when you introduce external disruptors into that network?
Okay. So I think what you would need to have some initial estimate. So you need to have kind of a link by potential transcription factors, or I mean, it depends on the disruption. So it's maybe a mutation introduced by a virus in the transcription factor. Yeah, then you just need to have enough expression data, I think, and it should be in two different groups, one with and one without the viral infection, and then you should be able to compare those two groups. If it's some totally new transcription factor that is introduced into the genome, I'm not sure if for example, a mutation that is introduced by the virus that causes a new transcription factor binding site. I have no idea if this is actually possible in biology, but then that would be more tricky because you don't know, so you don't have a starting point.
Thank you. Another question that was asked is about the in the case of the single sample networks in glioblastoma patients, where the single sample networks built by taking into account just one aggregate network inferred with all patients' expression profiles?
Yes, that's correct. Yes.
Thank you. Nice talk. I have a question. Do you think it is possible with Panda and Puma to model the effect of non-coding single nucleotide polymorphisms?
Yes, I think that should be possible. Yes, it would be a slightly different. I would probably do it with Puma. So yeah, but then, I guess it depends on how many SNPs you want to look into. So then it could, for example, be looking at SNPs at promoters, right? But then you have a quite long. So it depends a bit. I foresee this to become really difficult computationally. So if you want to look, I don't know, if you have known GWAS SNPs you want to look into, that's maybe a bit easier. So then you only have a few, and you can just link them and then look into the gene expression data. That would be very interesting. But if you would look into any SNP, it might be a bit more complex to do that computationally, or maybe you can focus on specific genes or specific pathways that you think might be interesting. That may be also an option. But it is still a bit computationally intensive to run these networks. And if you have lots of SNPs and then lots of targets, the networks are going to be really, really, really big. So just, yeah, I'm not sure if we're there yet to analyze those then with our current computers.
Thank you so much.
¿Alguna otra pregunta? Okay. Well, Marieke, thank you. Thank you very much. Really wonderful
Thanks a
talk.
lot. Great questions. Thank you.
Insisto, si alguien está interesado en platicar con la doctora, por favor, póngase en contacto conmigo. A los estudiantes que les gustaría charlar una hora más o menos con la doctora, estamos planeando una reunión a las 12 mañana.